Such excitement today. It is a rare day. It's a wonderful day, and it's really awesome to hear from Dr. Trivedi. Tell us a little bit about what you have done here.
So thanks a lot. Thanks, Steve. And Steve is being kind. He is with Dr. Potter, my partner, in this whole Big Southwest Node initiative. And in fact, we, in our resubmission, when we applied for a renewal for the Big Southwest Node, actually strongly argued and made it our major focus as the new stimulant abuse problem that we are now, everybody is beginning to recognize. But we had, and this study dates back to starting many years back. And Steve can do this presentation as well as me or better because he's been in the middle of all this for his entire career. I'm going to really give you top level results, but maybe also primarily open up for discussion for us, because I think in some ways this is the glass half full, half empty. The bottom line for methamphetamine use, as we all know, is there is nothing approved by the FDA, not for lack of trying. There have been profoundly large numbers of clinical trials with very little minimal effect sizes or no effect sizes with huge placebo response. And people forget that the basic reality for methamphetamine use disorder is threefold. One, it is actually not, it's common, not as common as something else, but still very much a big medical problem in the United States. The second is there is a huge presentation variability. Some people with meth use it over weekends or days, occasionally based on their interests. And a lot of people use it much more frequently, and some use it all the time. So there is a huge presentation variability, and then there is a huge treatment variability, outcome variability, so that some patients get better and some don't. And so not unlike many other medical illnesses. And so therefore these legion of previous trials, unfortunately, didn't really answer the question of what works for meth use. So we had to be mindful of that as we were designing it. That was one of the things we were very careful about, as Steve mentioned, and we'll get some active participation, hopefully. We had, unlike some other efforts, we wanted to convince CTN to do this, so their answer was, this has not been done before. And our sort of muffled answer was, if it's already been done, we are not interested, but we didn't say that. We said, okay, what will it take? So they agreed to let us do a small pilot trial that gave us some indication that there is some signal, but that, you know, as pilot trials go, it really helped us see the feasibility. But we had the same challenges that we had in mind. So we embarked on this study called ADAPT, and really, basically, it's oral bupropion with injectable naltrexone as our rationale. Our aim primarily was to assess efficacy of extended release injectable naltrexone. Product insert says every 4 weeks. We actually went to the FDA to get dispensation to give it every 3 weeks. Not a real profound question, but nobody had done that as a randomized efficacy trial, primarily because if you look at the pharmacokinetics of naltrexone injectable, at the end of 3 weeks, even before that, but definitely around the end of 3 weeks, the blood levels drop. And so we wanted to give this a fair shot. So we said we're going to do it every 3 weeks. And we used a high dose extended release version of oral bupropion, 450 milligrams, and used it as a combination. So we had a very clear placebo-controlled trial. I'll talk a little about the nuances of the specific adaptive study design we used. And secondary outcomes, as usual, were assessing safety, because one of the things people were interested in finding out is when you give this high dose and this short every 3 week naltrexone and oral bupropion, does it lead to significant adverse events in this meth use population where people have not used it? And then assessing secondary efficacy for other substance use disorder outcomes, depression symptom score, especially because we had bupropion as one of the treatment arms, and quality of life ratings. So one clear recognition that all of us need to go in with is Steve and I and Jennifer have spent our lifetimes doing clinical trials. You want to make sure that you give the population you sample the best chance to work that you're going after. And then you want to ensure that the clinical trial is very carefully designed to answer that one question. And there is a big tendency on the part of everybody to ask, why not all the other 99 questions? And I can think of the other 99. And as soon as NIDA funds us to do those, we'll do all those 99 studies. But this one was purely designed to assess efficacy in this population. So the clinical trial design really ended up being the most fun and the most challenging. And a lot of people kept on telling us, not to take it lightly, it won't happen. But nevertheless, what we decided to do was to use something called as SPCD design. It's sequential parallel comparison design. And it's basically a double-blind placebo-controlled trial. The sequential parallel comparison design is really orchestrated so that you have two phases, one stage one, that is, and the stage two are both mirror images in terms of duration. So in this trial, we had 6 weeks each of stage one and stage two. And both of them are placebo-controlled trials. In the first stage, you have active medication versus placebo. And the developers of SPCD design, Maurizio Fava at MGH, UT Southwestern is what I was going to say, but he didn't go to the good institution called UT Southwestern. So he's at MGH. He's a good colleague and friend. He's wonderful. So he does, with Pensina and a number of his colleagues at MGH designed this trial. Developed this design, where you then end up with a disproportionately higher number of people randomized to placebo in the first arm. And then at the end of 6 weeks, both responders and non-responders to the combination arms continue for another 6 weeks. But the active ingredient for the randomized control trial is placebo non-responders that get re-randomized to either stay on placebo or to the combination medication. So in effect, what you have is 2 phases. The first phase is a comparison of this combination versus placebo. I assume you're able to see my arrow. Yes? Okay.
Yes, we can see. Yeah.
The second part of it is this second arm of the combination versus placebo. They have recommended and they have used it in other clinical trials with different weights because this is a different sample from this. So their breakdown is a 0.57 weighted for one stage and 0.43 for the second stage. I'll talk to you about that as we go forward. So it ended up being basically a 12-week medication phase, and visits were twice a week. They got an injection every 3 weeks and oral medication every day. More about the study design discussions will come at the end, but I think that the population we were very focused on ensuring that we have a very good design so that the outcome criteria that determined who goes into re-randomization was maintained blinded to all investigators. So nobody was actually given the information that the primary outcome measurement for stage one was urine drug screens obtained in the 4 visits, that twice weekly visits for stage one, week 5 and 6. And the same thing for stage 2 at week 11 and 12, urine drug screens obtained provided the data for the primary outcome measurement for both the stages. That information was blinded to the investigators, so we had to do a significant maneuvering to directly deal with all the 9 IRBs to give them the data about when these outcomes will be determined and how the randomization will work, but maintain the blinding for the investigators. And in fact, that is the reason we did not go ahead and published the design paper before the study was completed. Then the second large component was to decide, make sure we chose the right population. And you can imagine for an undulating disorder where if you just used somebody, samples where they used only on weekends and you did urine drug screens on Tuesdays and Thursdays, you might get a false positive, false negative result. So we were making sure that the self-reported use of meth use for those who entered was at least more than 18 days in 30 days. That means in any given week, they had to be using it several days, if not every day. Luckily, we ended up with a sample I'll show you with a mean days of use of methamphetamine 27. That means these patients, 403 patients that entered, not many of them were actually able to stay away from methamphetamine for a day or two at a time. They met criteria for DSM-5 criteria for moderate to severe methamphetamine use disorder. And then the major other aspect of the trial was that they had to have at least 2 of 3 urine drug screens positive within a 10-day period. So you can imagine from, again, the pharmacokinetics of methamphetamine, that if you have 3 urine drug screens positive within 10 days, that means you're using it more days than not, or almost all days. And then they were medically and psychiatrically otherwise healthy. Doesn't mean that they were healthy, but they were at least stable, and they were not in any other addiction treatment. So this randomized controlled trial, unlike a lot of what is done in CTN, did not actually formally add or include any of the psychosocial interventions. People have asked us about that. Just to be very blunt in the beginning, we were not saying psychosocial doesn't work. It's just that the study was designed to test this intervention, and maybe subsequent studies can add other things if they want. So this is a sample we ended up with, about 2/3 male, not surprising, 71% white. That means our 30% of the population was people of color, which was wonderful. We were able to basically accomplish the task of not having a purely white population in our sample. 60% had some education beyond high school. This is actually interesting. That tells you that these are people who got into this later in life and were at least able to do some college, if not more. Half of them were never married, but there were about 40% of these patients were actually employed. So again, this is a very interesting sample. The baseline meth use was 26.7 plus or minus 4 days in the 30 days prior to randomization, and a third of them were baseline IV meth use. There were no significant differences in clinical demographic factors for both the stages. And I'll keep on repeating this issue of stage 1, stage 2, because that will come in stark review as you start looking at results, because we wanted to make sure that not only people entering stage 1, they were balanced in their sample, clinical characteristics, etc. But when you do this re-randomization, you didn't want a biased sample. So they were also still, there were no significant differences there. So there was no bias that inherently crept into the sample, even for stage 2. So the analyses, as I mentioned, primary efficacy outcome measurement tool was meth negative urine drug screen results for all the participants. A responder was defined as somebody who provided us with at least 3 out of 4 negative urine drug screens out of the, so at least they had to have at least 3 of the possible 4 they can provide at week 5 and 6, and again at week 11 and 12. And so the stage 1 evaluation period was 5 and 6, stage 2 evaluation period was 11 and 12, and then there were other safety events measured. The way the statistical and analytic approach works for this is basically looking at the comparison between placebo and combination at stage 1 and again at stage 2, and providing them with 2 different weights, and I'll show you that. This is the sample we got. So we had, as I mentioned, a disproportionately higher proportion of people randomized to placebo in the first stage, and then placebo non-responders were then re-randomized to either continue on placebo or get the combination. Placebo non-responders were continued, and drug responders, medication responder, as well as non-responders were continued for the 6 weeks. The key comparisons are this and this, the oranges are, I am a little color challenged, so whatever the color is. These are the primary outcome results. So in stage 1, we had primary outcome available in close to 75 to 83%, 68 to 83% of the population. And I think that if you were to ask me or waterboard me and say what really was the key ingredient that helped us actually prove what we had embarked on, I think this is it. We were able to increase and improve on retention rates and data availability in this trial with all the care we put into the random implementation of the trial. That some or a large proportion of the previous studies had not been able to accomplish. And I think all of you, the junior investigators, I can tell you, having spent a lifetime as a clinical trialist, what most people make a mistake is they design an elegant question and even come up with an elegant study design, but then don't get their hands dirty into the weeds and make sure that whatever you decided as your study parameters, that they are implemented to the best of its capacity. And I think that fortunately we were able to accomplish that, and that may have gone a long way into helping us get the results that we got. So it is a population that has basically in stage 1 a grand total of 3.4% placebo response. Somebody saying something? No. Okay. So you got 3.4% placebo response. So you can immediately know that this is a severe, severe, severe illness where people are not flocking into placebo response. And the combination response rate was 16.5% in stage 1, and then 1.8 versus 11.4. The effect size 0.11, leading to a number needed to treat of 9. So this is the bottom line for the study. This is probably why we were able to convince the editors of New England Journal to take it. Nobody has ever shown this. There has been no similar randomized controlled trial results. And the number needed to treat of 9 is comparable and in fact better than the number needed to treat that is seen with things like smoking cessation. Analgesic effect number needed to treat is 9. And you can see this, our primary outcome number needed to treat was 9. Naltrexone to prevent relapse to heavy drinking is 9 to 12. In abstinence and opioid use disorder with naltrexone, number needed to treat is 8. Now, look at that. I think that, again, tells you that we are in the ballpark with number needed to treat, showing nobody should be able to then argue that this intervention is better than doing nothing. It is comparable to all these other interventions. I'm going to take a pause, Steve, to see if there are any questions, or I don't know. Because the rest of it is trying to actually provide more context. Anybody have any questions?
we've been monitoring the chat, but if you want
Okay.
to just kind of contextualize the number needed to treat equals to 9. How do you know your patient's going to be number 9 and not number 7 or 2? How does a clinician use that number?
So I think the way to look at that, Steve, is twofold. One is I don't think we have too many biological biomarkers in our field, and for that matter, even in other chronic medical diseases, including hypertension, diabetes, et cetera, where you would be able to a priori predict whether your patient is really the one who's going to get better. So that is true for most chronic medical diseases and their treatments. Otherwise, we would only need one antihypertensive and one treatment for rheumatoid arthritis and one treatment for lupus, right? So that is the one thing. The second part is, I think subsequent studies after having proven that this works may be worthwhile doing by doing some biomarker-driven studies where we look at Curie activity, craving changes, et cetera, with these treatments as a stimulus and then put them on the treatment. That we currently don't have.
A follow-up question to that is, do you have a sense of what the number needed to treat for Naltrexone and Bupropion separately for methamphetamine use disorder from prior studies was?
I think I can't remember, but the effect sizes were so small, probably 20, 25, I don't know. And even those are really flawed studies, so I wouldn't actually hang my hat on those.
And before you get into some more, a question came in asking, can you talk a little bit more about the decision for doing the SPCD design and the thought process that led you to that decision, as opposed to standard clinical trial?
Go ahead. Sorry, I interrupted.
No, just as opposed to a clinical trial, a standard RCT, 2 groups. What? Go ahead.
So I think and Steve can chime in because he has heard these. And he is even better at it than I am. I'm just a poor outcomes researcher. And I do not want to spend 5 years doing something where the outcome is already destined to then doomed to fail. People have done regular randomized controlled trials and failed. There is a whole lot of actually uncertainty and assay sensitivity problems with randomized controlled trials for medical illnesses like depression. We've done depression trials a lot. And with addiction, where the phenotype variability is often, and the treatment outcome variability, if they are large, then you have the false signaling that happens with placebo response that is so common, that these previous trials convinced me that doing another randomized control trial is like waiting for Godot and maybe you will get lucky and get a positive trial, but there is no way of actually orchestrating a very fair assessment of the efficacy if you just continue to do the same. And if you look at actually other fields like oncology, rarely now does a pure placebo-controlled randomized controlled trial. They do routinely adaptive study designs because the phenotype is actually such. This is not infectious disease where you can do that. And so therefore, I think, Steve, you know this, and we talked extensively, no point in letting, wasting money from NIDA on something that then you just have to depend on luck. And this high placebo response in a lot of previous trials really scared us, that we would then end up with a trial. Failed trials are really a waste of money, as you all know, because then you can't conclude that the drug didn't work. All you can conclude is, give me more money to do another trial.
True. A question, can you just revisit the issue of the psychosocial platform that was used on this trial and your thoughts about whether, I mean, because I actually have thoughts about, or some questions about the question of the type of medication, behavioral therapy that was here wasn't like a support therapy as much as it was medication adherence. But do you think as this moves into, say, primary care settings, that docs would be well advised to have the focus on medication adherence as they're doing this study to get that, to maintain that high level of response that you saw here?
So short answer is, I think that in our disorder, especially meth use, so I think that that education of the patient about the importance of staying on it, and then follow up with patients regularly about helping them overcome the challenges and barriers they're going to run into about staying on the treatment should be an important integral part of care. But the benefit in real practice now is going to be that when we started, we were doing a clinical trial, and there was no evidence that this this works. And so we had to work a little more. Now that we know this works, I think the task for primary care physicians could be easier, but it still doesn't take away from the responsibility that for our disorders, we should educate and follow up and provide some some behavioral support in order to ensure that they are able to stay on it to attack their challenges and barriers.
Great. If you want to continue, I think the questions will continue to accumulate, and we can come back as we get closer to open talk at the end.
I think one of the inherent questions that is out there, and I think you didn't directly go to it, but I think it is implied, and that is, could we then improve or should we be thinking about adding bona fide psychotherapy to do this in order to reduce their meth use further? And the short answer is absolutely. I think that maybe gives me, I'm answering my own question, but it gives me a chance to really attack the nihilism in our field. Only in our field we actually argue about this. Most of medicine doesn't sit down and say, oh, is it medication or is it psychotherapy? We should be thinking about it, because that is how most of chronic medical diseases get better. Can you imagine a diabetologist saying, you know, I'm just going to give you insulin. I don't want to find out anything about your hemoglobin A1C, and my nutrition nurse doesn't have to talk to you. They don't have that debate. We do. So I think it's adding psychotherapy to this as an intervention and then trying out what is the added benefit is a worthy study. And any of you young investigators hungry to get an R01, maybe go ahead. The pattern of urine drugs screens in this one is actually quite interesting. If you look at this is people, individual patients, and their third week of in the stage 1, to see how the pattern of urine drug screens goes. Red is those who have a positive urine drug screen. Green is those who have a negative urine drug screen. And you can see just from the pattern, while week 5 and 6 was the evaluation period, you can start seeing that for those on the medication combination, this signal of improvement is beginning to show up around week 2 to 3. You look at this pattern on week 3, you can actually begin to get a much stronger sense of who is likely to get into a response status. This is for stage 2, same issue. You're seeing a little, obviously, as you saw in the earlier slide, the total number of people getting better in stage 2 is smaller than stage 1. We were obviously challenged to make sure that this was not just a kind of epiphenomenon. So we did a whole lot of different aspects of sensitivity analysis in many different ways. So we looked at the number of covariates, site, age at onset, baseline number of use days, IV use, DSM-5 criteria, number of days of cigarettes used, baseline treatment effectiveness assessment score, and visual analog score. And none of these made a difference in the fact that the combination was superior to placebo. There are subgroup analyses that we were obviously all wanting to do, and we looked at it. I want to caution all of us to recognize that as soon as you get to subgroups in this sample size, while 403 is the largest meth use randomized controlled trial, it still is a small number as soon as you start breaking it into subgroups, so you want to be cautious. But one thing is very obvious from this: under 40, over 40, in terms of race, ethnicity, if you look at the effect sizes, except for African Americans, where the sample is 48, you see that the effect for superiority of the combination over placebo is consistent across all the subgroups. And even where the sites, none of the sites or any of the subgroups, you see placebos doing better than the combination medication.
Mhm.
So it gives you real confidence that the effects are real, gives you some signals of what we should be thinking about in the future. Individual site differences in multi-center randomized controlled trials is the rule rather than the exception. So I'm not surprised there are some differences, but no single site showed that placebo was better than drug. And because there is mathematicians who are our peer reviewers, they always want to do more. So we used many different approaches. For example, as you can imagine, if we are only looking for those people who give you a negative drug urine screen for the primary outcome measurement, what it automatically indicates is those days where they didn't show up, they did not give us a negative urine drug screen, and therefore missing urine is by definition in the primary outcome is basically assumed to be positive urine drug screen, right? Because they didn't give us the negative. If you impute the missing urine drug screen as positive, it is the same, right? So this is that. If you imputed a missing urine drug screen as negative because you suddenly got very happy with the patient, even then you get the same benefit for the combination versus placebo. That is generally not something we would imagine in our field, but that was something in terms of sensitivity analysis. We looked at those who gave us the full, complete data, and for that group, so therefore it is not like we selectively only got people who gave us negative urine drug screens. There also you get the same benefit. If you adjusted the weights given to the two stages, remember it was 0.537 and 4.3 in the design based on SPCD design. But if you did the equal weightage, you still get the same additional benefit for medication combination versus placebo. This slide is actually quite fun to look at because it gives you a visual of the urine drug screen results in stage 1 and stage 2 for our ITT population. And you can see that the available urine drug screen results are clearly indicative of the negative urine drug screens for responders are much better than those for non-responders. Secondary outcomes, we looked at treatment effectiveness score. That is the proportion of 12 urine drug screens. Remember, in the original primary outcome, we basically looked at 3 out of, or at least all 4 of the urine drug screens, week 5 and 6, and again at 11 and 12. But they could have given us a total of 12 urine drug screens, in which case the proportion wise, again, you get benefit for the combination versus placebo. Timeline follow back, which is obviously the gold standard for quite many studies, although we went with objective measure with urine drug screen, but if you just went with self-report with timeline follow back, results don't change. There's also reduction in craving for the, that was measured with the visual analog scale. Craving reduction is much higher for combination versus placebo. Cigarette smoke abstinence, same. Reduction in depressive symptoms, again, more benefit for the combination versus placebo in both stages, and the combined effect was. So all of the secondary outcomes are positive. Quality of life measured through the treatment effectiveness assessed, the PEA scale. Is Walter on? He'll be happy. So that showed the same benefit. So I'm going to go very quickly to safety monitoring so we can then open up for questions. Safety monitoring, as I mentioned earlier, there was a lot of concern originally when we designed it. The DSMB was vigilant very actively. Serious adverse events, we had the one thing about this trial that even the reviewers at NEJM, the good news with the NEJM paper is all of you who have written them, and you know, I've had several of them, is that they are very good at making you sweat. So I actually, the benefit of that is you can dream of your data. You will never get a question that you have not actually looked at the table. There are some studies where you have to say, let me look at the table. In this one, you don't, because we had like 5 re-reviews and 4 page proofs. So 4 people got a serious adverse event on placebo, and 3 people got serious adverse event on, hold on. So 4 people got a serious adverse event on placebo, and 1 got a serious adverse event on the combination. Would feel like placebo is actually worse, but that is, just remember, it's 4 out of 294, and this is 1 out of 109. So it is, the rate is the same. So no major difference in serious adverse events between placebo and the combination. And none of them were actually life-threatening or led to death. There was actually 1 seizure on placebo, go figure. And so no difference in stage 1, again, no major difference in stage 2. In stage 2, as you can see, for re-randomized samples, now they're both equal or proportionally very close. So therefore, the numbers are similar here, 4 and 3, but nothing, again, of any significant impact. They were hospitalized. Adverse events, no major differences. There were significant numbers-wise, but they were mainly mild with some moderate, but very few severe adverse events. So the combination worked, and all of the secondary outcomes listed here were positive for the combination. I think that the real people who really deserve credit for this study results are these sites. Every site, every node, every site PI, and most importantly, the participants who gave us their time, energy, and faith in us. Because remember, when we went there, we said, nobody has done this. Do you want to do it? And so we can't thank them enough. And special thanks to, obviously, Steve and Jennifer for doing this with me. But Tom Carmody, if he's around, he's the master statistician who never wavered when I said we are doing an SPCD design. Everybody around was basically, it took me 3 months to actually convince them, but they finally relented because, I don't know, Steve thinks because I was not going to go away. All right, guys. This is a true pleasure for our node. Thank you all, and more to come.
Awesome. Thank you, Dr. Trivedi. A few questions came in. So let's run through them quickly, and then we can unmic maybe, and people can ask themselves directly. But what are your thoughts about working with bupropion plus mirtazapine that also has shown a little bit of a signal about using those as a combination, perhaps with the extended release naltrexone? Do you worry about serotonin syndrome from that kind of a combination? What are your thoughts?
So I think that the question of I think the results tell us the following, in my view. One is we should probably be thinking about next step directions with this combination to figure out how to study it in a little less severe population, in real clinical practice settings, in implementation models, et cetera, and really focus on this as much as we can, because here we have evidence. The second line of studies, in my humble opinion, would be to try to answer the question earlier that somebody brought up, and that is, are there specific patient groups for whom this is the best thing? And so therefore, behavioral as well as biological marker-driven studies that help us enrich the sample and do a little more precision treatments. The third would be to look for combinations that include one or the other of this combination with something else, which is the question that came up in terms of naltrexone plus mirtazapine. Personally, I think that is worth trying, but I am not yet sold that it is likely to be profoundly better than this combination, but that's purely non-scientific personal judgment. Should be studied. There may be other novel pharmacotherapies that may be worthwhile doing. And there may be, I think, time to start thinking about whether a two-step or a two-sequence study design is a better solution than actually doing another combination. But fortunately, I don't write checks for NIDA, so all of these proposals are actually worthwhile. And if any of you want to submit a grant, Steve and I and Jen will be very happy to help.
Thank you. Dr. Oliveto, if you can unmute yourself, you had a question. Dr. Oliveto is our co-person in charge of this journal club. So if you want to,
sure.
Yeah.
Sure.
Go ahead.
Hey, really, really fabulous. Thank you so much, Madhukar, for your for the information. You know, for many, many years, there's been a lot of dialogue back and forth about how do we look at outcomes for our trials. And, you know, some might use a treatment effectiveness score. You used an interesting outcome of responder based on the last 4 urines. So what were your thought processes? How did you come up with that? And because I did notice that you really didn't see much of an effect when you looked at something like treatment effectiveness score and that sort of thing. And how is the field moving in terms of how to better, you know, have consistency across trials, that sort of thing? Just, I mean, for me, this is a bigger discussion that I was really curious about in terms of how you came up with what you did and how this may move our field forward in terms of thinking about more consistent outcome measures.
So I think this is an interesting issue, Allison, that you bring up, because I think that we get sometimes there is so much of tradition about this, because I think that we come at it, all of these outcomes come at it from the whole AA model that once sober, always you have to be sober for you to be imagining that your life is better. And the alcohol field has actually moved on. I come at it also, as you know very well, Allison, from the depression world, because that is where I did a whole lot of my research there, where improvement in symptoms and response, et cetera, has been used as a very good outcome and has been tied to improvement in lives and function and quality of life, et cetera. And we have not done that as much in addiction. And as you've heard, Dr. Volkov has repeatedly talked about harm reduction. And this group of patients we had was going to be very severe meth users. And the reason we may not always have all 404 urines is not because they're using drugs, but because their actually lives are chaotic. And so we said, if we can improve somebody's life enough that they can give us 3 negative urines in 2 weeks out of 4, that's a big improvement. So that is how I came up with it. And fortunately, the DSMB slapped me around a little, but did still at the end of the day let me do it. And I think you can see this. I mean, for completeness, just so you know, and I don't know if Tom is on and can show it, but we also looked at the sample, defining it as all 404, Allison. And for that group defining it as all 404 also, we did find a benefit for the combination versus placebo that was statistically significant.
Okay.
But that was not our primary outcome.
Well,
I personally do believe that
Well, and I know in the past the FDA kept having a very high bar, like the last month abstinence, that sort of thing, or they kept shifting it around. And I was just curious if the CTN and NIDA have had those types of conversations with the FDA for medications trials, just to, if they ever came to some sort of compromise on what one can move forward with potential FDA approval, depending on that, the outcome.
So I think, Allison, first, I'm in Texas, as you very well know, so I'm not imagining the FDA to be the mecca of science, right? They follow the science, and I think that the way, at least to my sort of, whenever I've had discussions with the scientific community and the FDA, the issue is often chicken or the egg. They want to wait until the scientific field accepts an outcome, then they'll do it. So I think this is a first step. We are going to have a conversation with them, with the treatment development group, David McCann and Kurt Rasmussen and I, and we are having that conversation. But I have to imagine a little bit of it is going to be an iterative process. They're open to thinking about it. They are saying, as the scientific community already accepted it. And as you know, we are still, if you go to CPDD or ASAM, not enough people are talking about 3 out of 4 urine drug screens as a decent outcome, right?
I actually have another one, but I know another quick question, and that was regarding, so it looks like you report on the first 6 weeks and then a second group of 6 weeks. Did you see a continuation of improvement or at least stabilization in the first group that got the combination therapy through the full 12 weeks? Because it didn't look like those responses were added to the second. So I was curious about that.
I think that's a good question, and we've been so terribly kind of mired in addressing the NEJM things that we have not yet looked at that question, but that's immediately we'll do it.
Okay. Sounds like another paper.
That is, so Steve and you have taken on this mantle, so I appreciate your pushing us on papers. Thank you.
We have a few minutes for other questions.
There were some other questions in the chat that folks may want to jump on and just ask themselves, right, Steve?
Yep.
And just so any of you, if you don't know, the reason for this journal club was because we recognized that Dr. Potter's tweets about stuff is actually very exciting. So please join her club if you have not already joined.
Give me more lead time next time, and we'll get you that bomber, the Zoom bomber, not a bomber, not a bomber.
I think they are all still trying to go to Washington. So
I've got a question for you if you have a moment. I'm really, as an addiction psychiatrist, of course, I've known many patients to be on bupropion and have methamphetamine use disorder. and sometimes not in that order, sometimes in that order. And I don't think it's uncommon for people to use or misuse bupropion. Did you track at all the route people did take the bupropion, and do you have any hunch, does that have any effect on the signal response that you saw?
Just so I understand the question, route, we gave them oral pills. Is that what you're?
Of course, and I realize you use the extended release, which are more difficult to crush.
Okay.
But that doesn't stop everyone. Did you ask, did people crush it, inject it, snort it? And if so, any idea, do you think that might have had a role in the signal response you did see?
So I think, John, it is possible they did, but we were very vigilant about the whole process. So we actually used something called AI Cure. I don't know if you know, that is a software that has AI in its name. AI, so that if it requires you to put the capsule in your mouth, and it captures the facial expressions and the swallowing, so that at least on every occasion that they did not purposely not give us that information, we actually have that information, so that they used it exactly just as we prescribed. That still leaves those occasions when they basically didn't allow us to see it, but that was very small proportion. So I'm not saying it didn't happen. I'm just saying that the likelihood that was a huge number was very low.
It's true. And that part of medication adherence is something I think that's kind of implicit in your question. We can talk more about, because I do believe that there is something important about talking about people's pill taking. It's especially as part of it's the other part of the conversation that patients have with their providers, the prescription pad, and then what they're doing with their medications. So
yeah, more to come. I think that pill taking behavior oversight on our part was actually, sometimes we worried it is too much, because we gave them a smartphone, or they had to have a smartphone. Every time they actually ingested, that automatically sent a signal to our database, and that actually transferred to a dashboard. So our coordinators actually had real-time dashboards. And so you can imagine, once you as a coordinator have something on the dashboard, you have to then take action and all. So, and as a result, every time the participant came back, there was conversation with them. So Steve is right. There was directly and indirectly a lot of education and hand-holding about ensuring that they're taking their pills.
Okay, guys, I have the unenviable task here of representing Allison and me to say thank you so much, Dr. Trivedi. This has been tremendous. I also think you've set a very high bar for the Journal Club, but you know, I think one of the things that I've learned about this Journal Club is it always is interesting. So you bring quality, other days I'll bring the comedy. It'll work out. So it's good.
You know, Steve is understated, but I think one thing that does come through, Steve, and there is one more step we have to go through ourselves as the node leadership. But we may bring back to this group the publication plan for this study, so that people who are interested in either partnering with those papers that are already on the docket, but more importantly, Allison, if there are new topics that people want to get out as papers, Mena is already writing one. There is the paper now.
Yeah, where is that paper?
So we can bring it back here.
Great. So just a shameless plug, we meet here every Wednesday, second and fourth Wednesday. You are welcome to join us regularly. It's always energetic. We have standard practice is half-hour presentation about a journal article and a half hour on a works in progress. We have sign-up sheets. We have people lined up, and we look forward to continuing an energetic conversation. I know today is a little bit different format because of this unusual and wonderful thing of a medication for methamphetamine use disorder. And so we had to have more of a presentation format than an interactive one. But in general, we have a lot of interactivity on this page, and we look forward to having you all join us.